Okay, my name is Mark Purdue, and I'm the other co-moderator for this session. Dr. Nichols wasn't able to join us in person, but I would like to thank our speakers for their presentations and invite them to participate in the panel discussion. And just as a reminder, we encourage our audience to submit questions and comments via the chat box located below the webcast video. Please include your full name and your affiliation with your questions. So just as an initial question, I'd like to throw to the panel, what do you see as some of the most promising opportunities for advancing our understanding of environmental exposures in pediatric cancers? And conversely, what are some of the biggest obstacles to this research that we need to find a way to overcome?
I'm happy to start. Thanks for the question. I mean, I think from my perspective, the kinds of definitive data that we can get from the biospecimens that Joe and Lauren talked about are critical for our ability to really do a much better job with understanding the relationship between environmental exposures and cancer, especially for children, and especially given the fact that we can now be able to get these definitive measures looking at specific critical windows of exposure, I think is key. Something that we really haven't had much opportunity to do until some of these new methodologies have really come about. So to me, that's the biggest opportunity that we have in that regard.
Yeah, I would totally agree with that. And in the past, we've looked at exposures one at a time, and I think it's really the combinations that are going to bring answers. And now we're able to use these multi-omic platforms, such as the genetic platforms and the exposomic platforms that Lauren talked about. So the biggest challenge will be taking this huge amount of data, and might be artificial intelligence. I know that was talked about earlier, to actually sort through all that data and find specific answers, or find the wheat within the chaff, so to speak.
Okay, go ahead, Dr. Patrick.
Yeah, so I mean, I obviously agree with those comments. It's talking about the assays that I work with, and I think that really, we're talking about being able to measure sort of these broad environmental exposures, depending on what type of sample type it is. But we can also collect things like alcohol and tobacco. We can also measure cortisol. We can measure caffeine. So the ability to collect some of these other risk factors that may be working together with some of these environmental factors all in a single assay, as these technologies have really been driven to be able to measure things at very low concentrations and really small volumes, as well as quantitative approaches. Some of the quantitative assays now can measure multiple classes of chemicals quantitatively, up to 100 or so in a single assay. And so I think this is really something that could be leveraged for identifying these factors in combination.
Thank you all. We have a question from an online participant. Is there any data on environmental toxin exposure from schools? For example, school renovations, construction of schools adjacent to old school sites, or toxin exposures from aging buildings, et cetera. And I might broaden the question to ask whether there are risk factors, either established or suspected, that sort of co-localize to a school environment. Any thoughts from the panel on that?
I can start again, and my other folks can chime in here. I saw that question as it was posted, and have been thinking about it a little bit as the other talks were going on. I think two primary exposures that can come, especially from the aging buildings part of that question, obviously asbestos exposure from older buildings when that was still used for construction, and lead specifically in paint, probably more so a little bit in terms of neurodevelopmental outcomes rather than cancer per se, but lead is still sort of on the list as a possible carcinogen to humans. And I think once we know a little bit more about its potential contribution, we may be able to find out more. I am not currently aware, and Joe or Lauren, if you do, please speak up, of any studies that have specifically looked at exposure to, demolition or construction activities relative to schools. I know there has been some concern over the use of certain materials for playground composition, especially old recycled tire rubber materials. And we know that the exposure to some of the chemicals used in the development of rubbers is carcinogenic to humans. But I think this is an opportunity that we have to continue to look at these potential broader exposures that children might be exposed to during early childhood that could contribute to development of cancer specifically.
The only studies I've seen on schools tend to be motivated by a perception of a cancer cluster. So for instance, the Fallon cluster, the schools there were studied extensively for, a jet fuel pipeline that ran underneath the school. There's another cluster investigation where supposedly the wiring in the school was all done poorly and there was all kinds of EMF exposures. Another one would be a new school where formaldehyde exposures coming out of building agents was an issue. So I think both new schools and old schools have their issues. But I've never seen a definitive, I think Melissa mentioned a school earlier where there was a perceived brain cancer cluster, but when they did the population statistics, they couldn't prove it on the registry level.
Thank you. Another question from an online participant asking specifically about the evidence surrounding exposure to ethylene trioxide, which is used for medical device sterilization, wondering whether ethylene trioxide or other maybe ethylene oxides have been linked to any pediatric or adult cancers.
So yeah, I'll take this one too. And if others have more information, please feel free to add. I saw this one as it was posted as well and quickly pulled up. So we did mention ethylene oxides in general in the presentation that I gave. It is a group 1 carcinogen according to evidence that IARC has reported on. It's primarily been associated with breast cancer and lymphomas in adults. Very few, if any, studies have been conducted to look at the association with children. I think given though, as the participant pointed out, the recent evidence and so forth around this particular, medical device sterilization facilities, I think we have some opportunities to maybe start some geospatial analyses with in the areas of these particular industries and see if we do see anything that might point to an increased risk for any of the pediatric cancers.
So
Thank
I know
you.
that
Go ahead, Lauren. Sorry.
yes. So I know that there are some studies that have looked at TCE, occupational exposures, and metabolomics in order to identify some of the pathways that are altered to sort of link those to different health outcomes, including cancer. So there's a few studies like that have looked at what are the, to try to make that the linkages between the exposures and either intermediate cancer outcomes or cancer itself.
Thank you. Michael, a question directed to you regarding a comment you made about air pollution. Perhaps you saw this question from Sharon. So why do you think air pollution might impact outcome of disease or prognosis, survival, as opposed to the risk of developing the disease?
Yeah, no, that's a great question, Sharon. Thanks for posting it. I want to just clarify, I'm not saying it's necessarily opposed to contributing to risk. but I think we have an opportunity to look at the potential contribution of exposure to high levels of air pollution on outcomes. We've started to do that locally, especially for our leukemia participants on our different epidemiologic studies. And we do see an impact of living in areas of high pollution on poorer outcomes, both immediate treatment outcomes as well as longer term survival. So I think some of the hypotheses surrounding this is that exposure to some of the chemicals that might be involved with air pollution may have other detrimental effects on your health that would eventually impact your overall survival from your cancer diagnosis. I think also we can think about this being as a potential proxy marker for some other exposure that groups of individuals who are also at increased risk for having exposure to high levels of air pollution may have. So I think there are a number of ways that we can look at this, but I think it's important to start thinking about it, especially, I'm here in Houston, Sharon with me here as well. So I think there are a number of important environmental exposures that we commonly have now that we really have the opportunity to start looking at in relation to outcome. And I see Catherine also pointed out in the chat, air pollution has been reported to increase the number of respiratory infections in childhood cancer survivors compared to their non-cancer individuals. I think one other point I wanted to make is that exposure to air pollution may also create a sort of increased level of an inflammatory state within individuals due to the chronic exposure to particulate matter or whatever it may be. So that may also contribute to poorer outcomes.
Thanks, Michael. I have a couple of questions regarding investigating gene-environment interactions. So one asking, what role do genetic variants in cytochrome P450 genes and perhaps other related genes involved in detoxification systems, what role does such variants play in modulating the intensity of environmental substance exposures and by extension cancer risk? Or perhaps what may be known or suspected based on evidence to date?
Yeah, I could take that. I think there have been many studies on looking at pharmacologically important genes and genetic variants on risk factors, mostly in the occupational setting that I'm aware of. And this is something we've looked at in the past with benzene exposure, for instance, and butadiene exposure, where we know that there's a clear exposure to the population. And then we've chosen, different like cytochrome P452E1 or quinone oxidoreductase genes that have known functional polymorphisms and have found that they do impact the effect of the environmental agents. Now, in the settings of childhood cancer, where we've already heard today, there's a short latency between birth and the disease, and they're not going off to work to get these occupational exposures. They are, however, exposed to cigarette smoke and other factors. And as I showed in my that Manhattan plot, where maternal smoking during pregnancy and childhood leukemia at risk, we were kind of hoping we might see some of those, those P450 genes or other metabolic genes. We didn't in that analysis. What we did see are some genes that have been involved in lung cancer, which gave us some comfort in knowing that we're looking at, we are seeing some known genetic risk factors for addiction to tobacco, which kind of made sense. But really, we haven't. I'm still hoping we reemerge metabolism as a genetic risk factors within metabolic genes in childhood cancer risk. I think we have the technology to assess every gene in the genome now, and so we're looking at this more agnostically rather than as candidate genes like we did in the past. So I think our studies need to get larger, and our environmental exposure assessments need to get a little bit better to maybe see those emerge again.
Thanks, Joe. There's another question regarding genes, maybe more from a GWAS context. But the question is, how do the statistical associations identified as gene only or gene environment interactions relate to conclusions about disease causation when those genetic variants are found? And of course, these variants typically, when we first find them, these findings are tag SNPs that are perhaps included on a SNP array or imputed. So, question to the group.
So yeah, childhood leukemia there's probably over 15,000 kids who have been assessed by GWAS studies to find these gene-only effects. And now there's about 23 different SNPs associated with childhood ALL. And we can put the effect sizes of each of those SNPs together into polygenic risk scores. And we do find children who have extremely, they're on the high end of these polygenic risk scores, indicating a very high risk to leukemia, similar as if they had a predisposition genetic syndrome. Now, that's completely without thinking about environment. So we're just very in the early stages of looking at gene-environment interaction. There are very few gene-environment interactions in cancer, even in adults, that have been identified successfully because they require very large studies. And I think the one I presented today with maternal tobacco smoking, it's the first one I think I'm aware of in childhood cancer. And we still don't, we only have enough power to really see one that's a significant gene environment interaction. Now, I don't think that's going to solve, leukemia. We're not going to prevent, we've identified some individuals who have a variant who seem to be more highly susceptible to the toxic effect of tobacco. And if they had that knowledge, it would be information that would lead them to quit smoking if they knew they had it. And I suppose maybe ultimately that's the kind of information we'd like to get back to the public, that such susceptible people do exist.
There's a follow-up question since you raised polygenic risk scores. What about polygenic risk scores in the context of genetic variants as found to be associated with susceptibility to environmental factors? Or so, for example, sensitivity to UV damage. Is there any have such data been applied to further understand the etiologies of childhood cancers? And I might extend that to questions of applying Mendelian randomization type analyses, leveraging GWAS data to understand environmental factors. Joe, I hate to put you on the spot, but
I feel like we're 10 years away from
okay.
being that sophisticated. And yeah, I'm not aware of that, but if anybody else would like to chime up, I think, yeah, if we had, for instance, a biomarker of UV damage or maybe circulating cell mutational signatures that we could assess in healthy people, and then also look at UV damage and then do the genetic study, we might be able to do something like that. I don't know if Dr. Alexandrov is still on. He might be able to comment on something like that.
So thanks, Joe. Moving to a different area, it's been one common theme across the presentations has been the value of using dried blood spot cards for use in epidemiologic research, molecular epi research of childhood cancers. Are there? This is maybe more from my understanding, what are the barriers to using this kind of resource at the moment? For example, is it required to have signed consent from parents of the children associated with these cards, or is the opportunity to potentially extend data linkage-based studies to incorporate dried blood spot cards as a sort of follow-up, or are we more confined to working within studies where the participants have been directly asked for consent to participate?
It really depends on the state. Some states require active consent for use of the cards. States like Washington, Texas, Ohio. Other states long ago established an opt-out procedure where parents could opt out of their child's blood spot to be used for future research. But even in those states, they need to be used completely without personal identifiers. So to work closely with the state registry people so that if the cards are supplied for use in research, they are just supplied in a coded manner where they couldn't be traced back to an individual. The states that did establish such methods include California, Michigan, New York. So it really varies. And I think in just about every case, there are restrictions on data sharing and transmission of data, especially on small numbers within publications and so forth.
Thanks, Joe.
And sorry, Mark, I'd just like to add to that it's highly variable across the states. And we've seen some states even get to the point where, like in Texas, we had to destroy a lot of the spots that we had stored for research use and implement new procedures, as Joe mentioned, for actually now it's an opt-in procedure. So as everyone on the call can imagine, that creates some biases with the sample of the parents who have taken the time and effort to opt in for the allowance of their child's blood spot to be used. So it's great when we have states like California and Michigan and New York and some others who have taken steps to be proactive in that regard, because it's a tremendous resource, especially for pediatric cancer research. It's sad that we can't get to the point where it's universally useful for all populations, but that's where we are at the moment.
Thanks, Michael. Maybe time left for one or two more questions. One question. So clearly, research on pediatric cancers is hard. It's hard to do. Requires, in many cases, a lot of effort to develop studies of reasonable sample size to, draw conclusions regarding associations with reasonable statistical power. And I'm wondering, how can data sharing be more broadly implemented and leveraged to facilitate and accelerate research on pediatric cancers? Certainly, there have been collaborations and consortia, established to date, and you may want to comment on those. But do you see ways for us to, further facilitate collaboration and data sharing across investigators to move the science forward?
Joe, you want to start?
Oh, yeah, sure. Yeah, definitely. And I think it's come up today already, pediatric cancers being rare. The St. Jude's, they may have the biggest numbers, but even they need to reach out and to Europe and other places to really get enough numbers. I think consortia is is definitely the way to go, where individual studies can be pooled, perhaps at one location. We're having some issues with our the Childhood Leukemia International Consortium. There's a genetic study based at Logan Spector. I know he'll probably talk about that later, so I shouldn't talk about it too much. But it's an effort to at worldwide get all the childhood cancer cases, their genetic, their GWAS data, and pool it at specific locations around the world. There's 3 locations. And part of that, the reason for having 3 locations is because it's difficult to share everything in one place, but there are means can be can log into a computer distantly to work with data, and under an IRB that doesn't allow direct sharing back to that other university, you can still find a way to work with the data. So thank goodness for modern technology for allowing that.
Thank you, Joe.
Minute
Go
to
ahead, Lauren.
Yeah, so I think, obviously the importance of, pooling these samples in the small sample sizes is and the consortium that were discussed. I think, one thing that was mentioned earlier was really talking about the bioinformatics side, right? Where this is really evolving and evolving pretty rapidly. So maybe data that was collected at a certain point a year from now can be analyzed in a new and innovative way, which utilizes much more statistical power, or you're able to mine that data to find new environmental chemicals or, the structurally similar cousin to something that may be important. So, having these resources available for this advancement to really reanalyze, I think it could be a valuable way forward.
Thank you, Lauren. And with that, I think we'll conclude the session. So I'd like to thank the speakers for some wonderful presentations and a really stimulating discussion. And I'd like to introduce Dr. Alejandro Sweet Cordero, who will be moderating the next session, Strategies for Communication about Environmental Exposures and Cancer Risk. Thank you.